Good morning, good afternoon, and good evening, everyone around the world. I'm honored to welcome you all to this webinar interview with Jennifer Chow, the Chief Executive Officer of the Chimeric Therapeutics. I'm Echo Hindle-Yang, the CEO at MSQ Ventures, a New York-based cross-border advisory firm specialized in life science. Jennifer Chow is a cell therapy pioneer, an expert with extensive experience in developing and commercialization FDA-approved CAR T-cell therapeutics. She has led the commercial development for more than 15 pipeline cell therapy and has a proven track record in the biotech and pharmaceutical field with over 20 years of experience. Throughout this webinar, we will explore Jennifer's expertise and the Chimeric's approach to cell therapy development. We will touch upon their progress in clinical trials, their strategic plans for the future, and how they are addressing the challenges facing the biotech industry. Let's welcome Jennifer to our webinar interview. Jennifer, thank you so much for joining us today.
Hi, Echo. Thank you so much for having me. It's a pleasure to be here speaking with you.
We are excited to learn more about your journey and gain insights into the dynamic field of cell therapy.
It is an incredibly exciting field and a very young field, Echo, right? So much progress and so much activity going on in such a short amount of time.
Yeah, it's funny you say that young, but you already have so much experience in this field. So could you please tell us a little bit about your journey and how you become the cell therapy pioneer and expert in this field?
Yeah, I would love to. You know what? I will tell you, I think a lot of it was curiosity and luck, probably, Echo. I actually have been in the pharmaceutical biotech industry for 25 years. I started my career very much in what would be considered kind of a, maybe for some, a very traditional way. I actually started off in the field in pharmaceutical sales. And really, that's how I learned and began, and then just started to work my way up and wound up working in marketing for a number of years. I've been very fortunate to work at very, excellent companies like Roche, where I learned oncology. And then I wound up at a company called Celgene. And so Celgene obviously now has part of BMS. But in the early days of Celgene was very much an innovative pioneer itself, and spent a number of years at Celgene working on Revlimid and Vidasa, and then had this opportunity at Celgene to join a very early phase development team that was looking into cell therapies. And so this is back when what is now known as Abecma, the multiple myeloma CAR T approved by BMS, was in its early days in a collaboration with Bluebird Bio, and it was known as BB2121. And at the time, Echo, I can tell you, nobody really believed that CAR T cell therapies had a lot of potential. I think people thought they were interesting science, but there was a big question of whether they would actually provide real patient benefit and whether you could commercialize them. And so funny enough, my first role at Celgene in cell therapies, I was actually told, well, it may not be full-time cell therapies. We may have to give you other things because there may not be that much to do on cell therapy. So, I'm sure you can guess that what actually happened was exactly the opposite of that. And the cell therapy world just exploded, right? There was early data in multiple myeloma that was incredibly transformational for patients. And that alongside the data that everybody was seeing in, lymphoma B-cell malignancies that was incredibly transformational for patients just made this field take off. And so for me, I was so fortunate, I think, to be in the field in the very beginning sort of this generation of the cell therapies and really be able to learn from the growth of Abecma and Breyanzi at Celgene. And then I actually had the opportunity, after I spent a few years leading out the global commercialization strategy at Celgene, to move over to Kite. And there I was able to take on a role to lead out global marketing and operations and analytics, and very much work on that early stage pipeline and what Kite was developing, as well as on, what was already moving forward for patients with Yescarta and Tecartus. So I very much sort of look back and really think that I was just very lucky in some ways to be in the right place in the right time. But also, I think for me, cell therapies are just incredibly appealing, and I love them because they have such a profound impact on patients, and they are yet an incredible challenge every day. They're new problems to solve every day, and so you certainly never get bored. So that's really been my journey through cell therapies up until Chimeric. And so I was at Kite when I had the opportunity to join Chimeric. Chimeric is a clinical stage cell therapy company. We're actually an Australian founded company, and we're listed on the ASX. And I had an opportunity in the very early days of Chimeric. Our founder, Paul Hopper from Australia, had actually licensed one technology and was looking for someone to start to build out the company. And this was just a phenomenal opportunity to be able to take everything I had learned at Kite and at Celgene and to really be able to develop a pipeline of assets that I thought would be transformational for people, and really to be able to move things forward to the patients that actually need these therapies the most. So I've been here at Chimeric for about 2.5 years. Very proud of the fact that we have a very small team, Echo. I think we have an incredibly experienced cell therapy team, but we also have some incredible assets that have great promise for patients.
Congratulations on all the success and the milestone. Actually, Chimeric, I've been like for such a short time, you have been making so much progress. I think also just listen to your journey. I think that can take days to really get into and learn so much from that. Let's know when the Chimeric can be successful in such a short time. So as a CEO, and you also have been just like you said, excellent companies is the industry leaders. So what do you learn from there? I think you must see something missing. That's why you want it's better, right? So what's your vision for the company?
Yeah, it's a great question. And I can tell you a little bit of what I thought I was seeing a number of years ago. Cell therapies were so exciting, Echo, when they first came out. There was, as I'm sure you know, this explosion of cell therapy companies popping up everywhere, and an incredible amount of investment moving into cell therapy. It sort of the 2019, 2020, 2021 timeframes, it was just this continual growth in cell therapy. And I think in some ways, what that actually enabled was a bit of a focus away from actually the patient at the end of the story. What I think happened a little bit when we had so much excitement and so much investment in the space was we had a lot of people and a lot of great scientific ideas. How could we improve these therapies? And people really went out and started to really develop technologies and really look at how could I make the next greatest technology. But what people moved away from was actually developing products and assets. And I think, the technologies are wonderful to develop, but the challenge with that sometimes is they can have very protracted timelines for commercialization, right? You can spend years and years developing a technology and trying to optimize it before you ever think about putting it in a product. So one, I kind of felt like we were doing so much innovation in science and so focused on just making tweaks to technology that we were losing a little bit of sight of actually building therapies that could get to patients as well. So that was sort of one thing I think I was seeing prior to my time coming to Chimeric. The other, and I'm sure you've seen this as well in the cell therapy space, was that so much of our energy was concentrated in very small number of targets and a very small number of disease areas. So, as we were thinking about looking for how do we show our technology is better. Everybody was in the CD19 target antigen space, right? Or the BCMA space. And even to this day, there are over nearly 600 assets just against CD19 alone, right?
Yeah.
So the other thing I was really seeing was that there was this focus on CD19 for B-cell malignancies and BCMA for multiple myeloma. And then all of a sudden, you kind of went from 600 assets being developed to 20 assets that were in different, other things. So I think there sort of became less innovation around trying to target new assets, new targets, different types of cancer. And then I think the third thing that really happened alongside all of that was, as we had this explosion of companies, Echo, there actually became, a real challenge of having experienced people fill all the roles in all of these new companies. And so suddenly a lot of companies, I think, were challenged around bringing in people that had the right experience and expertise in this space. And although, there are thousands of people with phenomenal backgrounds in small molecules and monoclonal antibodies, that experience doesn't necessarily automatically translate to cell therapies. So for me, I reflected on sort of those three big things as I thought about building out the strategy for Chimeric. So what we've done is we've really decided to stay focused very much on novel antigens and novel areas. So number one, our assets are first in class. So our CAR T cell therapies, we are targeting antigens and disease areas that are very unique. The second is that we've made sure that we have a very, very experienced team. So all of my team has cell therapy experience from multiple companies, because I think that actually helps our diversity of thought, but lots of experience we have. And then the third is that as we think about developing our assets, Echo, we really focus on what we call commercially conscious development. And very much that's about developing an asset that we believe is actually a commercially potential asset for patients. So we are not so interested necessarily in developing technology platforms. We're really interested in developing therapies that can get to patients. So that's really how we've kind of come about building the company.
Oh, that's so actually, from what you're sharing, I think how to quick access by their patients and treat the patients actually is the key to drive their whole process and the strategy, right? So you had so much experience in the commercial development for more than 15 peptide cell therapies. So can you walk us through the commercialization process and how you bring these therapies to market? I know this can be like this is $1 billion question, but maybe you can just summarize in a very high level.
Yeah, absolutely. Echo. You know what? I think it's important to think about patient right from the beginning. A lot of talks I've done and people that I talk to, they haven't actually thought so much about where the asset, what patient the asset's going in. So I think that's number one when you think about commercialization. Even when you're in a preclinical stage, thinking about what unmet need exists and what patient group you're looking at. And so that sets you up right from the beginning. And then developing a really strong target profile, product profile. So you really have a clear line of saying, my product needs to be this good for it to actually really help patients in that area. And so really that can really then guide your development, right? Because then you know exactly where you want your asset, what patient group you believe will benefit from your asset, and what bar you really need to show in terms of safety and efficacy to be successful in a commercial setting. So I think setting those things up really early, Echo, are really important in development for successful commercialization in the end.
That's really true, because the patient is the end user, right? You need to start with the end, the who are consumed, they're like be treated, and also the physicians who are going to actually use their drugs to treat the patients. So, and you have been working on the developing and commercializing several FDA approved CAR T therapy. So could you please share with us some of the challenges you faced because this is still, like you said, very young, right? So there's sometimes not so much like established rules, or even maybe you have to even advise the FDA group how to really like a regulatory, and so you can like shape the industry. I think you probably already have so much the experience to be the advisor, even not just to the company, but actually to the regulatory or the government. So could you please share with what's the bigger challenges you faced during the development process? How
Yeah,
did you overcome them?
exactly. I think in the very early days, Echo, when I was working on, BB21 and Breyanzi, and in the early days, some of the challenge was that this was such a young field. The regulators didn't really have guidelines, right? They didn't really know what was going to be important any more than probably us as drug developers did at the time. And I think that what that actually created in the early days was a really collaborative community where everybody was coming to the table and saying, okay, we need to be able to talk amongst, regulators, so the FDA, as well as company representatives, and really be able to work together. And I think that was such a great part of what allowed CAR T cell therapies to get approved. Since that time, over the course of the last, 6 years, I guess, since those early discussions and early days, the FDA has obviously done an incredible job of starting to put out all of the different guidelines and really continuing to stay abreast of all of the development in our space so that they can try and respond to some of the challenges we're seeing, right? I think, in terms of as a company thinking about taking something through regulatory, the regulatory pathway to commercialization, one of the really important things is to think about the CMC and the manufacturing. And, I think that's where people sometimes have stumbled or been delayed. And I think that is for everybody probably part of or one of the most challenging pieces to commercialization.
So that's how it's not really like a one side decision. It's not really like a government have the power to say how to do that, but it's really like collaborate, especially for the new area, right? Like how to influence and how to show their the heart to the patients. I think if that works for the patients, that it will be works. Yeah. So I want to.
Yeah. Sorry, Echo. Yeah.
Yeah. No. You please go ahead.
No. I was just going to agree with you. I think when you see therapies that are so transformational for patients, everybody wants to make sure that they get there.
This, I think, is really breakthrough, and we see so many like the disease we couldn't figure out, and now we have the hope. So next, I want to switch the gear a little bit. And so we know there's several different therapeutics like CAR T cell therapies and MSC and NK, and then the TLL, and even the gene editor cell therapies are well known there in the world. The sales of the approved CAR T are increasing, like approval. That is really means that is really effective for the patients, right? But however, we see the over the market, the CAR T cell therapies actually is not so many clinical trials going on around the world. We see there's 15 in China, and then there's probably not like a 2 in Europe, and then there probably 3 or 2 in US. So what do you see the landscape will be in the next 5 to 10 years globally?
Yeah, you know what? I think you're absolutely right. There was sort of this explosion of looking at all sorts of ways to develop cell therapies. And so the initial cell therapies were CAR T cell therapies that were autologous, and so very personalized to a patient. I think some of the inherent challenges of an autologous cell therapy led a lot of people to believe that the answer was going to be allogeneic, so these off-the-shelf cell therapies. So a lot of development work is in that space. And then it moved into exactly what you said, looking at other cell sources like NK cells or other ways to do things. And I think there is, an explosion of work being done in those areas. I think in terms of clinical trials going on, I think there are a lot of early stage clinical trials going on, Echo, with all sorts of different assets, different cell sources, different targets. I think what we're really starting to see now, though, in terms of those numbers that you were mentioning, is those cell therapies that are actually moving towards commercialization, right? So there was the initial group of the sort of 6 CAR T cell therapies that have been approved in the United States to date. But now we're actually starting to see the next generation come through, right? We're starting to look at potentially an approval for a TIL, which will be the first cell therapy in solid tumors, which is really exciting, right? Because I think we've all talked about how challenging cell therapies in solid tumors are. So I do think that what we're starting to see now is that next generation of cell therapies get through those important trials that would take them into registration. And interestingly, I think analysts have expected that this market over the course of the next 5 years will go from sort of being a $4 or $5 billion market to a $40 billion market. So we're expecting to see that commercial growth. And, you see that reflected in the recent announcements the FDA has made about how they actually have reorganized and are committing to growing the resources in the FDA in cell therapy. So I do think people are really, sort of getting there and also even gearing up for the future to be even stronger in the cell therapy space.
Yeah, actually, I still have many questions for you, like this area, but we have limited time. So I will save the question is, as a female leader in the biotech industry, I know a lot of people ask how you balance, but actually as a CEO, as myself, female CEO, actually I want to ask you, what are the advantages to be a female CEO, and what are the benefits for the company shareholders to have a female CEO?
Wow, that is a great question, Echo. You know what? Because usually you're absolutely right. You get the flip side of that, or what are the challenges? But I do think that there definitely are some benefits, right? I mean, for me, when I look at it, and you were sort of talking to balance, for me, actually, I have 2 daughters. So for my family, I actually love the fact that they have a role model that actually, inspires them to go on and to think that they can do all sorts of things. But for the company, I actually think there's a lot of benefits that female leaders can bring as well, right? I do think that there is definitely, we as female leaders have really had to continue to push and demonstrate our abilities and really show ourselves. And I think at every stage, very much, you really have to be able to show how strong of a leader you are and how strong you can be. So I do think there is definitely this perseverance and this determination and drive that female leaders have that is extraordinarily strong, because we probably have faced challenges as we've come up through the ranks. The other part of it is, I really do believe that female leaders tend to have a stronger sense of teamwork and collaboration and empathy across their organizations. And perhaps even in some ways, for when you're developing therapies for patients like this, for patients, right? A big part of what's guided my career is these patients that I have been very fortunate to meet along the way. And they've inspired me so much, Echo, right? And I think that is a big part of, I continue to think about, okay, this asset I'm developing today, how quickly can I get it out to that person that I met that really needs it? So I do think there's some great advantages in female leaders as well. But I'm sure you also would have some good insight on that piece.
My insight is I'm okay to cry to my husband because I'm a female.
Yeah.
But sure, all the male CEO, is that easy for you to cry to your wife?
There you go.
Thank you so much, Jennifer, for such sharing. I totally agree with you. That's as a female CEO, there's a lot of benefit, and also especially, I'm so glad to see more and more the female CEO come to our webinar. And so the next year we should really do the female CEO run the table. So next, please welcome you to share the present your company information on the pipeline. So we will continue the Q&A after that. Thank you.
Perfect. Thank you, Echo. Now, just give me 1 second here and I will share my presentation, and let's just make sure we've got all of this working here. There we go. Now, hopefully you can see that, the slides.
Great.
Okay, perfect. So thanks, Echo, and everybody for taking the time, not only to join Echo and I, but also to let me introduce to you, for those of you that don't know Chimeric Therapeutics. So Chimeric Therapeutics is a clinical stage cell therapy company. We were founded in 2020 by Paul Hopper, who is an Australian bio-entrepreneur who's actually founded numerous biotech companies. And we are listed on the ASX. And our focus is very much, as you see here, about bringing the promise of cell therapy to life for more patients with cancer. And this really goes to just what I was saying a minute ago to Echo, is that I've been fortunate to meet patients that were told that potentially their disease, their lymphoma, their myeloma was so severe that there was nothing else that would help them. And then they actually wound up on a CAR T-cell therapy. And then you hear 5, 6, 7 years later, they remain cancer-free. And so very much our company's goal and its vision and its mandate is to bring those outcomes to more patients with cancer. So that really is what we're all about. So, very excitingly, one of the things that's happened over the course of the last number of years is that we've been able to actually now start to say with confidence that the scientific data lets us conclude that cell therapies, specifically CAR T cells, can actually cure certain types of cancer. And so here, these are patients with leukemia. And these are patients that actually were treated in the very early days at the University of Pennsylvania and are now more than 10 years cancer-free. So very exciting, because we're first and foremost able to say that we've actually been able to cure these patients of their cancer. And not surprisingly, sort of as I was saying to earlier, that's come with this explosion expected in the market. There has been an incredible amount of investment in science and really exciting development going on in this space. We see in 2022 sort of the market being estimated to be about a $10 billion market. By 2027, analysts expect it to be over a $40 billion market. And so you really see the growth that is expected in this field being what we anticipate will be the fastest growing market in oncology. It will grow at a much faster rate than we believe that other types of assets, monoclonal antibodies, or those types of things, small molecules, will be growing. So a very exciting time to be in cell therapy. Cell therapy, though, like, I was saying to Echo at the beginning, doesn't come without its challenges. And so what we've really tried to do is we've tried to build our company around these 3 strategic pillars that we believe really allows us to position our assets and our company for growth and to be able to benefit the patients we're trying to bring our therapies to. So first and foremost, our assets are innovative assets. Our CAR T-cell therapies are first in class. Our NK cell platform, we believe, is a best-in-class platform, and we have the robust IP portfolio surrounding all of our assets. Our team has that very critical cell therapy experience and expertise. Everybody has a background in this space, and we've actually been able to demonstrate, right, and prove we know how to take things from very early stage development through commercialization and actually get them out to patients. So we've actually been able to show we can realize the value as we take our assets forward. And then the third pillar we call commercially conscious development. And for us, this is really about looking at developing product candidates that we can move through registration pathways to get them out to the patients that need them the most. We really have focused our energies and our efforts in patient populations with high unmet medical needs. And we've really made sure that our manufacturing, our technical operations, we integrate the development of that right alongside our clinical development to make sure that it is growing and it's scaled for commercialization at the same time as our clinical asset would be ready. And then we have really made sure that we've built, as a small company, a portfolio and pipeline that is actually synergistic. And so our NK cell platform really is there to enable the allogeneic development of our CAR T cell assets if we want to take it in that direction. So these are really the pillars around which our company was built. In terms of what our actually pipeline looks like, then our portfolio, you see here on the left-hand side are autologous CAR T cell therapies highlighted, and both of these are first-in-class assets. CHM1101 is a chlorotoxin CAR T, and it is targeting solid tumors, right now most specifically recurrent or progressive glioblastoma. CHM2101 is a CDH17 CAR T, and that is targeting gastrointestinal tumors. Our lead candidates there really are our lead areas are colorectal cancer, gastric cancer, and neuroendocrine tumors. Now, both of these assets are using first-in-class binders that have biologically relevant targets that are applicable to solid tumors. And that's really critical because we know that CAR T cell therapies have been challenged in solid tumors. So one really important thing is to make sure that the targets are very biologically relevant. And so both of these are. Both of these assets have novel construct designs that are really meant to make sure that they are able to overcome some of the solid tumor microenvironment challenges. And so very innovative designs. And with CHM1101, we are already in the clinic and we've already seen encouraging early data, which I'll share with you in a couple more slides, in that very difficult to treat recurrent glioblastoma space. So if I go to the other side, our allogeneic NK cell platform, we really believe has the potential to be best in class. CHM0201 is an NK cell platform, a healthy donor derived platform that's actually already been studied in a phase 1 trial for both solid tumors and blood cancers and shown some encouraging initial data, especially with one patient who had a complete response in acute myeloid leukemia. Our intent with that platform is to actually enhance that platform and then really use it as a backbone to let us develop allogeneic therapies with our CARs. So we see CHM1301, which would be an allogeneic CAR NK utilizing our chlorotoxin CAR T. Similarly, 2301 would utilize our CDH17 CAR T. The important part of this particular NK platform is it's already demonstrated persistence and expansion, which is one of those things that really has been very challenging for developers in the NK field to show. We see this platform technology allowing us to have multiple assets as we go forward, and I'll highlight for you a little bit of the early phase data we have already in this space. So first of all, I'll spend a little bit of time just talking about our chlorotoxin CAR T, CHM1101. It was designed to target glioblastoma cells, and so probably many people familiar with the challenges for glioblastoma patients. Unfortunately, its diagnosis, their survival is only predicted to be 12 or 15 months, and only 5% of patients actually survive more than 5 years after a diagnosis of glioblastoma. Patients in the recurrent setting, really generally are only looking at sort of 6 to 10 months survival. So this is a very, very deadly type of cancer, one of the worst possible. This particular CAR T cell therapy has been designed to be able to overcome some of the challenges and to target glioblastoma disease. It uses chlorotoxin, which is a very unique binder, to find the glioblastoma cells and actually bind to them. Now, chlorotoxin is interesting because chlorotoxin has been used historically as a tumor paint. So it's actually been used by surgeons to be able to show them where all of the glioblastoma cells are in patients. So when you actually think about it, it makes perfect sense to say, if you actually have something that can find and bind and highlight all of the glioblastoma cells, if you actually add the CAR T that can then actually kill those cells, it makes perfect sense to actually use that as a binder. Chlorotoxin, as I said, has been optimized in terms of hinge and costimulatory domains, and also in the way that the manufacturing process is set up around really maximizing potency and minimizing exhaustion for cells. So very much we are excited about moving this asset forward. This particular asset is in a phase 1A clinical trial already, and you see the design highlighted here. There were 4 dose levels design in this trial design. We've completed the first 3 dose levels and actually have started the dosing in dose level 4 already. With this, the patients are recurrent or progressive glioblastoma patients, and they receive 3 infusions, 1, 2, 3 weeks. What is very unique about this is the way that the CAR T cells are administered in this particular trial. So we know one of the other challenges with solid tumor CAR Ts is actually trafficking CAR T cells to the tumor site itself. These sites are directly administered into a patient's brain through catheters. And so in dose level 2, 3, and 4, patients actually have 2 catheters that are placed, an intertumor and interventricular catheter, and the cells are administered through those catheters directly to the disease areas. What we've seen with this trial already in dose level 1 and dose level 2, so this was very early data, is a very positive and encouraging signal. So keeping in mind that these were low dose levels, we already saw in the first 2 dose levels a 70% disease control rate. So 70% of our patients were achieving disease stability, and we actually saw that disease stability held for over 3.5 months. Now, 3.5 months may not sound like a lot if we were talking about certain disease areas, but with recurrent progressive glioblastoma, this actually is incredibly encouraging to us. And we saw this at the low dose levels with no dose limiting toxicities or really serious adverse events of note. So very encouraging, but very early stage data. What we're doing now is we're very excited as we're just in the process of opening a multi-site trial to finish off first and foremost dose confirmation of that highest dose level, 440. And then at the end of 2023, we'll actually have a key data readout where we're going to take a look at all of the data that we have from the phase 1A trial and do a safety and efficacy assessment to see if we believe that the signal is strong enough in this patient population to continue development. If it is, we are all set then to be able to move rapidly into part B of our clinical trial, which would be dose expansion, moving into 12 to 26 patients in dose expansion that would set us up nicely to move into a phase 2 trial that we believe would be a registration trial. So moving this forward, and certainly something to look forward to at the end of the year, is this data readout as we get there. So from there, I'll turn and I'll introduce you to CHM2101, and this is our CDH17 CAR T. This particular CAR T, again, another first-in-class asset, but designed for gastrointestinal cancers. And so CDH17 is something that's broadly expressed in GI tumors, and we are very much focusing as we begin the development of this asset in neuroendocrine, colorectal, and gastric cancer that has very high unmet need in that late line stage. 5-year survival for metastatic disease in colorectal gastric cancer, less than 15%. Now, this asset comes to us, we license this asset from the University of Pennsylvania, and certainly one of the most renowned cell therapy institutions in the world. The asset was worked on by the team at the University of Pennsylvania for over a decade. They've been optimizing the development of this asset. It is a 3rd generation CAR with dual CD28 and 4-1BB co-stimulatory domains, which we believe it will enhance its ability to overcome the solid tumor microenvironment. And then again, it has very much been developed around its hinge domain to maximize potency. And the length of that domain has been adjusted to really tighten immune synapse so that we have the best target engagement we can have with this asset. Now, CDH17, in terms of thinking about one of the key things to be able to overcome a solid tumor environment, is really being able to have biologically relevant targets. CDH17 is an oncogenic driver of tumor formation and cancer metastasis, particularly in GI tumors. And so we really believe targeting CDH17 is something that really will affect the proliferation of cancer in these cells or in this cancer type. What we actually have already seen is really incredible in vivo tumor eradication. The data here was all published last year in Nature Cancer as the cover story back in the March-April timeframe. Dr. Xinhua, the inventor of this asset, and Dr. June, who contributed to the development of the asset, were authors of the publication. What we saw in that publication was complete eradication of all tumors in 7 different types of cancer xenograft models. You see here just highlighted 4 of those. And we saw this tumor eradication with absolutely no relapse. And then importantly, because we know CDH17 is actually expressed on healthy tissue, we saw that tumor eradication without any treatment-related toxicity. So we really were able to take a look at the human and mouse affinity, and so be able to say, yes, we believe that the CDH17 binder recognizes both human and mouse CDH17 with similar affinity, so we could actually use the mouse models. We saw in the mouse models that the body weight loss in mice, there was no body weight loss and no damage to normal tissue at all, giving us a really strong safety signal in the in vivo setting. And additionally, no infiltration of the normal intestine by the CAR T cells at all. So with this preclinical data all set, right now what our team is working on is taking this to clinic as quickly as we can, because we really believe this has incredible promise for these patients with high unmet needs in colorectal, gastric, and neuroendocrine tumors. So lastly, Echo, I'll just touch a little bit on our NK cell platform and why we believe that is a differentiated platform and a really important underlying technology for our company. It's really with this about giving us the ability to move into an allogeneic space with our CAR T cells in the long run. So it's a platform technology that we can use across multiple different assets. It has broad applicability to more than 10 disease areas. And we've already been able to demonstrate encouraging phase 1A clinical activity here. There was a phase 1A clinical trial, and this actually was published last year in 2022 as well. This particular trial was done in patient population of colorectal cancer patients and acute myeloid leukemia patients, so both solid tumors and blood cancers. First and foremost, we were able to establish safety. Now, with this platform, it's a healthy donor platform, so the safety was extremely important to be able to establish without showing any graft versus host disease, and we were able to show that. So that was a very important point that we were able to look at. We were also able to show persistence. And so one of the things that have plagued the NK space is actually having persistence of the NK cells. Within this trial, we were able to demonstrate 28-day persistence of these NK cells. And then we were able to show expansion. And so we were actually able to do large-scale manufacturing from a single donor to be able to support this particular trial. So 3 really important fundamental things to be able to demonstrate with the NK cells. From an efficacy perspective, we saw a small efficacy signal in the solid tumor and the colorectal cancer patients of disease control. We actually believe that that is not strong enough, and so really do believe that it needs to be enhanced to be able to take to development in solid tumor patients. In the blood cancer patients, though, we saw this stronger signal of 100% disease control in the acute myeloid leukemias. And very notably, we had this one patient you see highlighted on the right, who actually had an early complete response at day 100, and who now is more than 2 years post her NK cell infusions with 0201 and remains in complete remission, which is, obviously very rewarding and exciting for us. So this asset is actually being developed now in 2 ways. For us, what we want to do with it is we want to enhance the asset really as a backbone for further technology. So we're taking that back into the lab and we're enhancing that asset. But the investigators in this trial also felt that this was actually quite promising. So we've also been able to move this asset into another new clinical trial, a phase 1B trial, in combination with an agent called vactisertib. And vactisertib is really meant to be able to enhance what we saw as the initial safety or the initial efficacy signal of the NK platform through tumor microenvironment modulation. This trial is open right now at Case Western Reserve University in Ohio. First patient was dosed in January of 23, and we're really excited to see what this combination can do for patients, both in the solid tumor space, again, in the colorectal cancer patients, and in the acute myeloid leukemia patients. And then this just highlights the ongoing development beyond that combination trial. We'll be looking at being able to add in IL-15 and a dominant negative TGF-beta receptor, which we believe will help us promote NK cell activation and further overcome the tumor microenvironment. And that will then give us a nice platform to be able to go ahead and develop CAR NKs with our CARs if we believe that that's appropriate as next step development. So with all of that, this is what our pipeline now looks like. You can see the green is the CHM-1101 trials I highlighted in glioblastoma. The orange is us moving that asset towards clinic for patients with GI tumors. And then the dark blue is really highlighting our NK cell development. And that's actually sort of as we are continuing to go forward. We believe that the pipeline then addresses these important high unmet needs in both solid tumors and hematological malignancies across our lead indications. We're targeting the colorectal gastric cancers, neuroendocrine tumors, and glioblastoma, and acute myeloid leukemia. And, you know, I would be remiss, Echo, at having any discussion of the company without highlighting the incredible team that we've been able to bring together. Small team here, the leadership team has over 75 years of experience in this space, coming from companies Kite, Fate, Juno, Artiva, so very much cell therapy focused companies. And we've taken 4 of the approved CAR T cell therapies through development to commercialization to really have the needed experience to be able to progress our assets to patients. We're also very fortunate to be guided by a world-renowned group of scientists and clinicians as our scientific advisory board. And so here, Dr. Lin, Dr. Bishop, Dr. Smith, and Dr. Maloney really have been instrumental in bringing those first generation CAR T cell therapies through approvals and helping guide their development. And this group have been incredibly helpful to us as we think about how to bring our assets forward for patients. So that really, Echo, gives you a sense of Chimeric and our assets, our pipeline. We believe the pipeline, you know, importantly has innovation and diversified pipeline with differentiated assets. We've already seen important early clinical promise. We really are focused on continuing to optimize that development so we can get our assets out to the patients that need them the most. And we believe that we have a strong team with the proven expertise to be able to actually drive those assets and get them through development to patients. So thanks, Echo, for allowing me to share that with you, everybody. I appreciate that.
Congratulations again, and so much the successful progress since the company have been just started in the last few years. So we collected some of the questions from the audience already, and so we probably wanted to go to more the deep, like the first question is from the audience is about your just mentioned about CHM 1101. So do you expect this will be changing the standard care?
Yes, and so CHM 1101 is targeted towards recurrent progressive glioblastoma. And unfortunately, Echo, right now, there is no standard of care for those patients because what we found is really nothing works very well. So very strong sort of as someone is diagnosed with glioblastoma, they'll come in and there is a strong standard of care. But upon recurrence, I think that there are a number of different options that people, you know, generally go, you know, can have, but really nothing that has emerged as a strong standard of care. So our hope is that this asset is able to show the efficacy needed to really become a standard of care for those patients that really need something.
Wow, so that is really save a lot, can like really save a lot of the patients' life and to solve the problem have been for so long. Still, we couldn't. So
Yeah.
you mentioned about how the manufacturing process designed to overcome the solid tumor challenges. So could you please go to more the details? I think this become like a very challenging for a lot of the CAR T therapy company, right? How did you and your team resolve this issue?
Yeah, I think it is manufacturing is definitely a challenge, but I think the other part, as we've seen, is that there are definitely challenges in CAR Ts being effective in solid tumors. And so there are, with our assets, different ways in which we believe we've optimized to be able to impact solid tumors. And with CHM 1101, our manufacturing is actually one of those ways. And so really that focuses on a proprietary cell selection process, right? That is sort of one part of it. And then what that does is it allows us to enrich for less differentiated cells. And that's an important piece because we really believe that that really improves the metabolic fitness of the cells, allowing for better proliferation and persistence of the cells. So very much it's sort of all stemming from being able to choose the right cells and then looking at how you manufacture those cells in a specific way to be able to really come out with in your product cells that we believe have good metabolic fitness so that they can really proliferate and have persistence when they are in the patient.
So how do you see that this process will be impacted on the ongoing clinical trial? So what are the key objectives and the endpoints of the ongoing and the planned clinical trial can be like a benefit from this process?
Yeah, I think what that actually does is we believe that impacts efficacy for patients, right? Very much. And so by being able to generate a product that is got better persistence and expansion when it is actually given to a patient, we believe that will then impact the efficacy of the product very much. And so, we're very optimistic based on the early signal we're seeing with sort of the 70% disease control in the low dose levels, that actually will lead to better survival for patients. So really, we believe that by having cells that stay around longer and are able to expand, that actually will directly contribute to better efficacy for patients.
That's very interesting, and also that is really like improve the treatment a lot. So let's go to the CHM 0201. I remember in your presentation you mentioned about the combination ongoing. You already have the combination, and you still looking for other combinations. So what's your criteria or strategy to select the other combinations?
Yeah, it's a great question. And so I think with CHM 0201, Echo, to be honest with you, we were a little surprised at the strong efficacy signal we saw, right? So these are non-engineered NK cells, 0201 cells. They very much in the first clinical trial, though, showed a strong efficacy signal, particularly in blood cancers. When we originally were looking at how we were planning to develop CHM 0201, we were actually planning to move it right back into the lab to develop it by adding that IL-15 and the TGF-beta receptor to further be able to engineer those cells for better efficacy going forward. But based on the strong signal that we saw in the clinic at the beginning with 0201, we moved into exactly that combination trial you were talking about. And so as we actually think about that, what we're looking for, Echo, is to see whether or not the signal with that combination trial in those patient populations is strong enough with the non-engineered cells to take that forward. And certainly if it is, we will absolutely progress that to next stages of development and move that asset forward for patients. But if it's not, Echo, I think for us, we are already working on 0301 that would take those additional enhancements into those cells to give us a stronger platform.
That's really makes sense, right? When you select, you want to find so you have to like open the door and to see which one is the most effective one. And so I'm just very impressed with the team in such a short time have been like expand such the collaboration or the combo, and that's really like amazing. So for your, actually another thing you mentioned about your core, the NK platform is very powerful. So, but that is another major challenge for the CAR NK therapy is persistence. So how do you overcome this challenge?
Yeah, you're absolutely right. Persistence is definitely something that has been a challenge in this space. And really, where other NK cell therapies have only been able to show a few days or up to 1 week of persistence, we are actually been able to show in that first phase 1A trial 28-day persistence, which we've been very excited about. We believe this is really due to the fact that we have a proprietary NK cell activation and expansion platform. And then we also have a very tailored flu-cy lymphodepletion regimen. But I think that piece around how we are actually activating and expanding our NK cells is really where sort of, that secret scientific differentiation comes. And that is really what is enabling us to get that NK persistence for 28 days, because we really believe that it is affecting the phenotype of the cells.
Mm. So in terms of this platform, actually, so how do you position the core NK platform and the next generation core NK platform in terms of the clinical development and the commercialization? So there's a several different direction you can go, right? You can develop your own, you can collaborate, and so which direction you are choose and why?
Yeah, I think that's a great question, Echo. I think there's definitely ways to go forward, developing in our own and in collaboration. And we're exploring both. So we have developed, obviously, a pathway for chimeric development, which has O201 being engineered with IL-15 and TGF-beta receptor inhibitor to give us O301, and then potentially then develop our CAR NKs from that platform. So we've built our own path forward, but we are also exploring different collaborations, and very much those collaborations really right now look like combination trials. So one is obviously the collaboration with the group that is doing the Case Western trial with Vactisertib. And there are a number of other discussions we're having now about other potential collaborations with that. So really, we are keeping ourselves quite open. Although we have defined the path for chimeric, we are keeping ourselves open to other collaborative opportunities as well.
That's great to know. That can benefit to the community.
Yeah.
And so for the next question, I just curious because I know the company started in Australia, and Australia have been very, the government have very been like a strong support to start the clinical trial there. So about, I just curious about your side of the clinical trial globally. How do you strategy, like where to go and where are they now?
Yeah, it's a great question. And you're absolutely right. Australia has been an incredibly strong home and support for biotech development. So for us right now, our clinical trials that are ongoing are in the United States. And really because these assets were invented in the United States. And so the first trials were actually done and are being done in the centers where these assets were all invented. So they were all invented in the US. As we go forward, I think there are multiple things we are looking to do, and one will eventually be to be able to bring these assets back to Australia, to be able to do clinical trials in Australia and in other countries. And then the other important thing for us is to be able to look at potential partnerships or collaborations to develop them in countries like China, where it's a little bit more challenging to develop as sort of a Australian or American company. So that is really the other part. So we really are thinking about the development globally in the longer term. It's part of, the next stages of development, Echo.
Thank you, Jennifer. Thank you so much for your sharing your personal journey and your expertise and also the insights about the pipeline and your strategy. So I know we have the limited time here. I hope we will continue this conversation and we welcome all the audience to submit more questions and to have the more deep conversation and help more the patients. Thank you again, Jennifer.
Thank you so much for taking the time to meet with me, Echo. It was great to speak with you, and thank you everybody for your time.
Thank you. So thanks everyone joining today's webinar with Jennifer Chow. So if you have any question, please feel free to reach out to our team or reach out to Jennifer directly. So we are pleased to co-host this webinar with BioAroundWorld, a platform that aims to streamline cross-border collaboration on global drug development by sharing real-life practical experience to achieve greater harmonization. Thanks everyone for joining us today. See you next time. Bye.